Hi, this is Max Rempel, and I will talk about approaches to genomic resonance therapy of cancer. And you can find my publications, full texts on dnaresonance.org, and also the videos, previous videos. I will be brief. It will be a brief introduction into the ideas about how genomic resonance can be used for therapy of cancer. So although sequence-specific DNA resonance signaling in the genome has been predicted over 59 years ago, experimental evidence is scarce. The RAP works by Montagnier, but that's about it. That's pretty much limited. And the details are still missing. Yet we were able to obtain evidence for it using computational approaches. And the main idea of DNA resonance is that similar electron wire structures and proton wire structures in the DNA, if they are similar, and if they, since they depend on sequences, the similar sequences would resonate and synchronize their conformations. So when they are connected through waves, they resonate and synchronize through conformations. And our contribution was that we were able to computationally predict electron wires in DNA structures and proton wires in DNA structures. So these are proton wires. And this is just shown the core of the DNA where the wires are located. It's called the base stack. And the proton wires are pretty sophisticated shape. So an electron wire is a little simpler, and we believe that they serve a biological function. So this is electron wire. And they are defined by the sequence. That's the key. So different sequences would have different resonance patterns. The range is very wide, ranging from kilohertz to an important band we think is 40 gigahertz. It's not proven, but that's predicted by models, and we have justifications why we think it is the frequency where the oscillations happen. So we identified specific resonance sequences and demonstrated that they enriched by the evolution, strongly enriched by the evolution, suggesting that they have a function. They are enriched in conserved areas of the genome, suggesting that they have functions and a function, and then nearby electron wire patterns are affecting the frequencies of mutations over the alleles of the mutations, suggesting that they are functional, that these wires are functional. That was the evidence. Now I will show several ideas for the use of DNA resonance for cancer diagnostics and treatments. They are based on lots of experience and the literature, but haven't been tested yet. So this is all at the idea stage. First, let's talk about diagnostics. A tumor, wires are inserted, thin wires are inserted, and impedance spectrometer measures the impedance spectrum. And for enhancement of the spectrum, it is the DNA purified synthetic short DNA sequences are used in their circuit to amplify the signal relative to a specific reference. So this would make this diagnostics more sensitive and help to identify the type of cancer using multiple DNA sequences. So the device would contain solutions of DNA in an array and compare the electric impedance spectra to different DNA solutions as references, and that would improve the diagnosis. For therapy, so again, synthetic DNA sequences will be screened for selective resonance-mediated killing of cancer cells. So these are cancer cells in a little cuvette. This is a DNA solution separated by a thin plastic barrier. It is activated by pulses of electric current, electric pulses. The DNA solution produces secondary induced pattern electromagnetic waves, which are sequence dependent, and different sequences are screened, tested against the ability to kill cancer cells using the secondary waves. So pattern electromagnetic waves produced by DNA when it is excited by electric pulses. And once the proper sequence is identified, we go to the next stage and we create an implant capsule containing that selected DNA solution, DNA resonator in solution synthetic. And the electric pulse generator is activating the secondary waves, which are yellow here, and they're spreading and telling the tumor to shrink. So here the capsule affects the tumor and commands it to shrink. In the next application, the idea is that the tumor survives mainly because it is signaling to the outside tissue that it is a useful organ. Usually it pretends to be a fetus and gives away the signals that it is a fetus. So the idea is to put here a capsule which would produce the waves signaling that it is a tumor and it should be killed. So basically inside the tumor, we put a source of biological waves, DNA waves, which would signal to the surrounding tissue that it is a foreign body which needs to be removed, signaling to the immune system. So for that, we synthesize candidate DNA sequences, load in capsules, implant in mice with model cancer tumors, and monitor growth and shrinkage of tumors, and select the best DNA sequence which works best for that. And this is it. You can find the publications and videos on the site dnaresonance.org. This is my email. And also you can subscribe, find the contacts here, and subscribe to webinars, which we do once a month. Have a good day.